Hello and welcome to this film which is all about important macromolecules. Now it's not a film about every important macromolecule, we'd be here for hours, but hopefully by the end of it you will be familiar with some important examples of addition and condensation polymers and their uses. You hopefully will have made a connection between this tertiary protein structure that we've been looking at up until now and the function of things called enzymes, and also will understand the similarities and differences between soaps and detergents. Now you might already know something about the difference between the way those two things work, but you might not know much about the structure of detergents yet, so we're going to have a look at that in a bit more detail. Okay, so we're going to start off with what we're calling here important addition polymers. There are so many important addition polymers, but the waste course wasn't going to have them all in it, so someone had to decide which the most important ones were. And the first of these is polyethylene or polyethylene. And as the name suggests, there are lots of ethene molecules that go together to make polyethylene. And so if you drew the structure of it showing its repeating unit, then you'd see that there were carbons going along in the chain with just H's hanging off, because if you were asked to break this chain down and show what monomers it was made of, then hopefully you just go along every two carbon atoms, put a double bond back in, and imagine that these single bonds weren't here. And there you go, there's ethene. But anyway, important things to know about polyethylene are that it's very, very commonly used in plastic bags and lots of other plastic bottles and packaging materials. Okay, we'll move on to the next one, which is ptfe, or polytetrafluoroethene, ptfe for short. Plumbers use this as a kind of joining compound on the threads that they screw together, but it's also very widely used in non-stick cookware and in lubricants because it's a very, very low friction substance. And hopefully, once again, if they asked you to say what monomer it was made out of, you'd imagine it with a double bond here and these two single bonds gone. There's poly, lots of, tetrafluoro, so 4 fluorines attached to ethene. Okay, so again, know its structure, know its name, know some uses for it. Now we're going to have a look at a couple of important condensation polymers. Again, there's lots of these, but we've narrowed it down to the ones that are in the waste syllabus. And first of all, we're going to look at nylon. Now, might be worth spending a little bit more time here on the structure of nylon. We've got two different kinds here. Okay, nylon 6,6 and nylon 6. Now let's have a look at why nylon 6,6 first of all is called a condensation polymer. Well, hopefully here you can see an amide group, rather like in a polypeptide or protein. Okay, and hopefully when you see this you kind of maybe think to yourself, oh well, there could have once upon a time been a hydrogen on this nitrogen, and maybe once upon a time there was an OH on this what was once an acid group, and these two things, when they joined together, they formed water in a condensation reaction. Okay, so if you can see that, then hopefully you can also see that the monomers that we'd have used to make nylon 6,6 would have been this diamine, so a molecule with 2 amines on it, and this carboxylic acid. Remember, this was a carboxylic acid before its OHs fell off. Okay, now why is it called nylon 6,6? Well, because there are 6 carbons here in the chain between the 2 amino groups at the end, and there are 6 carbons in this dioic acid, so this molecule with 2 carboxylic acid ends, and it has 6 carbons in its chain. So that's why we're calling it nylon 6,6, because both the monomers have 6 carbons in them. You could make nylon 6 in particular by using one molecule and one molecule alone, because this molecule, well, I suppose it's not a molecule, but it's the repeating unit of a polymer. If you imagine we had another NH here, then things are gradually starting to repeat themselves, then we'd have another CH2 and we'd have 5 of them, because we're talking about nylon 6, and then there'd be a carbon-oxygen double bond, and then we'd have the next nitrogen-hydrogen. Okay, so every one of these here, again, hopefully we're recognizing them quite quickly now. Here's an amide group, and this bond in here would have formed as part of the condensation reaction that would join these monomers together. Why is it called nylon 6? Well, once again, because there are 6 carbons here in the monomer. Everyone's come across nylon because it's very, very commonly used in lots of artificial fibers. It's also used in some plastics as well, but you don't need to know lots of uses for it, but it would be good to know the different structures of nylon and the fact that it's used in artificial clothing fibers. I suppose it's also useful to know that it's a condensation polymer, but then we know about condensation polymers already. And here's another condensation polymer, and this one's called polyethylene terephthalate, or in other words, PET. And you've probably come across this a lot because lots and lots of plastic bottles are made out of it. And you might be thinking, well, hang on a minute, you said that plastic bottles were made of polyethylene. Well, yes, but polyethylene isn't quite so clear when it's turned into a bottle as PET. So the manufacturers like the fact that you can see right into this bottle. And what's also true about PET compared to polyethylene is the fact that it doesn't leak gases as easily as polyethylene does. So if you put a fizzy drink in a PET bottle, it will keep its fizz for longer. Now here is the structure of PET, and again hopefully you're looking at it and seeing a condensation polymer. Why? Well, because there's these ester groups. Okay, so here's an ester group, here's the bond that formed in the condensation reaction. We can imagine hopefully that there was an OH here once upon a time and an H here, and that they fell off and made water when these two things joined together. And hopefully you can imagine that after this molecule here, which would be, I suppose, so let's just draw a line between the two monomers. So they would have been where the ester linkage formed. So there's one of our monomers, except it would have had H's on either end. So here we've got 2 carbons and 2 alcohol groups at either end. And this carboxylic acid that we used for this esterification reaction, which just happened again and again, so it was called a condensation polymerization. Bit of a mess now, my diagram, but hopefully you can see that between what was the carboxylic acid, or what were the carboxylic acid groups, you've got a benzene ring. So it's almost like benzoic acid, but it's got a carboxylic acid group at each end of its ring. So there's PET and a use for it. Now then, we're going to have a look at enzymes. Now, enzymes, if you didn't already know, are examples of protein molecules. The special thing I was going to say about enzymes, but the thing that we're most interested in about enzymes is the fact that they are catalysts. Okay, they're catalysts that occur in biological contexts. So they're catalysts that your body makes, for example. And just like most catalysts, we should be able to imagine the energy level diagram for a reaction with a catalyst. So that's the red line here, and with a catalyst is the blue line, without a catalyst is the red line. So what effect does the catalyst have on the activation energy, and how does that cause the reaction to speed up? This three-dimensional structure that we talked about and the importance of the shape of a protein comes into play particularly with enzymes, because the substrate, that is the thing that the enzyme is trying to catalyze in a reaction, will only be catalyzed by the enzyme if it can fit nice and neatly into the active site of the enzyme. And if you do biology, you might have heard about the fact that enzymes denature at different temperatures, and that's because the structure, this 3D structure, which remember we call the tertiary structure of this protein, will start to change as the temperature changes, and these intermolecular forces that are within this enormous molecule gradually break or form at the different temperatures. And so the active site will change shape, and the enzyme won't work very well as a catalyst at temperatures outside of its favoured range. Now, why do we bother using enzymes? Well, the nice thing about enzymes is that they're able to operate at very, very low temperatures compared to most industrial catalysts. Okay, so an enzyme can be used at about 37 degrees because that's the sort of temperatures that they like, which means that you save a lot of money if you're an industrial chemist because you don't have to go heating things up. Okay, so that's what enzymes are, that's how they work, that's collision theory, which we should already know about, and why they're special, and perhaps why we would use them instead of other catalysts. And now we're just going to finish off by talking about something that we actually know quite a lot about by now, and that is soaps and detergents. So here is a diagram of a soap, and we can see this big non-polar tail, okay, the hydrophobic non-polar tail, the water-hating part that likes dissolving in fats but not in water. And here's the water-loving end, the hydrophilic ionic end, which can form ion dipole forces with water. Now, if we compare that structure with that of a detergent, we'll see that a detergent, we've actually got a picture of a soap here as well. You can see this one down the bottom is just a soap. It's a very similar thing to that. But we've got two detergents here. And what's the difference between these and a soap? Well, they've still got the ionic part, right? This SO3- attached to a benzene ring, and then this big non-polar part to them. Okay, so just like soaps, their non-polar part is water hating. It likes dissolving in fats. Their polar part likes dissolving in water because it's ionic. But the advantage of them, as we should know by now, is that you can use them without them forming scum. So they work well in hard water, whereas soaps don't. What do you need to know about detergents? Well, just the fact that there is this SO3- group attached to a benzene ring, which is attached to this big long non-polar chain. Okay, so if you're wondering, out of all that stuff, which perhaps you maybe thought, oh, this is just some fairly unimportant molecules, which I've just been given some examples of, they are very important. They will be tested. There's lots of other very important ones that won't be tested. But what should you be able to do? You should be able to remember the names and structures of those two addition polymers that we looked at and the two condensation polymers. Well, there were three because there was two nylons. And also to be able to say what monomers they're made out of. So hopefully the monomers thing isn't new. You should know what an enzyme is and how it works as a catalyst, but that's just like any other catalyst, and why we might prefer it to what we might call an ordinary catalyst, and also the similarities and differences between soaps and detergents in terms of their structure. So now that we know a bit more about the structure of a detergent, we'll be able to talk about the way that a detergent differs from a soap in terms of its structure. We should already know about the advantages of detergents over soaps. Okay, so as I said at the start, the aim of this film was to become familiar with some important examples of addition and condensation polymers and their uses, and to make a connection between tertiary protein structure and the function of enzymes, and also something which was on the first slide but isn't on this one, and that was the difference between soaps and detergents. Hopefully it all makes sense. If you've got any questions or comments, then please feel free to come and see me, or just to post a comment on YouTube.